Hello friends, welcome back to this new video. I know it has been a long time, but honestly I was busy with exams and other stuff. But now I hope I can continuously upload videos and help you guys with physiology and afterwards with maybe with other subjects as well. Okay, so let us start this important topic. It's called as countercurrent mechanism. It's one of the most important topics in kidney if we have to understand how ADH, antidiuretic hormone acts, diabetes, insipidus, diabetes mellitus, and everything. So we should be familiar with this countercurrent mechanism. You all must have heard about this during your higher secondary classes, but here we are going to have a bit more detailed discussion of this topic. Okay, so countercurrent mechanism, as you can see on the slide, it consists of two parts. One is called as countercurrent multiplier, and other is called as countercurrent exchanger. Please focus on these words multiplier and exchanger. Okay, so multiplier means it has to do something with multiplication. Okay, in other words, if we are talking about kidney and if we are talking about countercurrent mechanism, definitely we have to talk about plasma osmolality and osmolality of nephric filtrate. Okay, so here in countercurrent mechanism, what countercurrent multiplier does is it multiplies the osmolality of urine, or in the earlier stages what we call as nephric filtrate. Okay, we are going to see this in the next slide. Just remember it, why it's called as multiplier. Nephric filtrate, okay, it has to multiply. This is the main thing, that's why it's called as countercurrent multiplier. And countercurrent exchanger, we'll get to that part later. So before starting the main discussion, there are three things that we should know. So in countercurrent mechanism, three things are very important and they are required for countercurrent mechanism. Without these things, countercurrent mechanism is not possible. Okay, so the first thing that's important is ADH. So, okay, ADH, as you all know, it's called as antidiuretic hormone. It's also called as vasopressin. It's also called as AVP, arginine vasopressin. Okay, there are three names of the same hormone: ADH, vasopressin, AVP, arginine vasopressin. As you all know, it is secreted in the supraoptic nucleus of hypothalamus and then stored in the posterior pituitary. Okay. Second thing that's important for countercurrent mechanism is it's called as vasa recta. Okay. Vasa recta. So we'll see this in the diagram what vasa recta actually is and how it helps in countercurrent mechanism. And the third component is called as urea. So these are three main components that are very important for countercurrent mechanism. One is called as ADH, antidiuretic hormone, vasa recta, and urea. Now let us try to understand how each of these component acts and how actually this countercurrent mechanism helps. And what is the purpose of this countercurrent mechanism? Again, the purpose of this countercurrent mechanism is to produce a highly concentrated urine. Concentrated urine. Okay, so that is produced with the help of this countercurrent mechanism. Now let's go to next slide. Okay, so this is an unlabeled diagram of nephron, as you can see. So I'm going to show some parts. You all must be well aware. This part is, so this part here is called as the Bowman's capsule. It's called as the Bowman's capsule. Okay, so this part here is the proximal convoluted tubule. This is the proximal convoluted tubule, and this U-shaped thing, it's called as loop of Henle. And the important, now this is important, this is called as the descending limb of loop of Henle, and this one is called as thick ascending limb of loop of Henle. Okay, this is the descending limb, this one is the descending limb, and here it's the ascending limb of the loop of Henle. This part here, this part, this whole part is called as distal convoluted tubule. Okay, so this is called as distal convoluted tubule, and here this is collecting tubule. Okay, now, okay, these are different parts of nephron and you all must have already studied these. Just to brush up the things, I again renamed them. Okay, now let us discuss the main thing. So this is the efferent arteriole. Here, this efferent arteriole, it divides into a bunch of capillaries which are called as glomeruli, and this is the efferent arteriole. Okay, so as you can see, blood, it actually once it passes through the efferent arteriole, it gets filtered through these capillaries. These capillaries, as you all know, they are called as glomeruli. So here, blood, it gets filtered, and whatever is formed here, this thing is called as nephric filtrate. It's called as nephric filtrate. This is called as nephric filtrate, and eventually, as it passes through different parts of nephron, as it passes from here, eventually it passes out. This forms what is called as urine. Okay, so this forms what is called as urine. Now, okay, let us try to understand what happens with this nephric filtrate and what is the role of countercurrent mechanism in all this. So, okay, the osmolality of this nephric filtrate, as soon as it enters into the Bowman space, the osmolality is 285 to 290 milliosmoles per liter. Okay, so this is the osmolality of nephric filtrate here in just as it enters the Bowman space. Okay, now as this nephric filtrate passes through the proximal convoluted tubule, some salts are reabsorbed and some water is reabsorbed as well. It means solutes are reabsorbed, solvent is reabsorbed, and at the end of the day, as the nephric filtrate leaves the proximal convoluted tubule, its osmolality again remains exactly same. That's 285 to 290 milliosmoles per liter. Okay, it remains exactly same. That means whatever reabsorption took place in the proximal convoluted tubule, it is called as isoosmotic reabsorption. That means equal proportion of solute and solvent got reabsorbed, so it's called as isoosmotic reabsorption. It's called as isoosmotic reabsorption. Equal proportion of solute and solvent got reabsorbed in PCT. So at the end of the day, again plasma osmolarity remains same. So here there is this nephric filtrate and its osmolarity is again 285 to 290. Now, as the nephric filtrate goes down through the descending limb of loop of Henle, its concentration goes on increasing. So at the tip of loop of Henle, right at this point, the concentration is very high and its maximum, that's 1200 milliosmoles per liter. Okay, its maximum 1200 milliosmoles per liter, that's maximum in any part of nephron. So that clearly means here it's say approximately 300. Okay, this is 300 and here it's 1200. It means during this part, it means throughout this part, that is descending limb of loop of Henle, a large amount of solvent reabsorption took place. So large amount of water got reabsorbed in this part, and as a result, and no solute got reabsorbed, so the plasma osmolality here became 1200. It became 1200 at this point. That means plasma osmolality got multiplied from from the value of approximately 300 to the value of 1200. So this part is called as the counter current multiplier. Multiplier. Why is it called as multiplier? Because the plasma osmolality, because the osmolality of nephric filtrate got multiplied in this part. And so which part of nephron contributes to the countercurrent multiplier? As you can see, so let me write it here. It is the descending limb of loop of Henle. Okay, so this contributes to the counter current multiplier. So again here it's 1200. Now as the nephric filtrate ascends through the ascending limb of loop of Henle and reaches the DCT, the osmolality is in the DCT is around 50 to 100 milliosmoles, and as this passes into the collecting duct and here, so once urine is formed, the osmolality of urine is around 800 to 900. That means it's highly concentrated as compared to what's present in the DCT. Okay, so this DCT here, urine is formed, which is highly concentrated. So how does this occur? So here we can understand from 1200 to 500 to 100, this part. So we can understand that a lot amount of solutes got reabsorbed. So here large number of solutes got reabsorbed, solutes, and because of that the osmolarity decreased from 1200 to 50 to 100. Okay, lot of solutes got reabsorbed, so that's okay. Now what happens from DCT to collecting duct? From DCT to collecting duct, the plasma concentration again increases to 800 to 900, and it's like a huge, it's almost 9 times increase in plasma concentration. And how does this occur? So here we have the role of other two important components of the countercurrent mechanism, which are vasa recta and urea. And actually all three components are important here only. First is the countercurrent multiplier, we already discussed that. Now these three, they play an important role mainly in this part, that's countercurrent exchanger, which is again a very important part of countercurrent mechanism. So let us try to understand what happens actually here. Okay, so this is the nephron. Okay, now it means the nephric filtrate from this point to this point has to undergo a lot of water reabsorption. That means lots of water should be reabsorbed. There should be reabsorption of very high amount of water for the plasma osmolality, for the osmolality of nephric filtrate to change from almost 50 to 100 to 900 at this stage, and the so that concentration of urine finally is 900. Okay, this happens because of the presence of this hormone, which is called as ADH, antidiuretic hormone. Okay, now before discussing what actually happens in this part of nephron and how countercurrent exchanger occurs here to form concentrated urine, let us discuss how this ADH acts first. Okay, so this is okay. If we magnify this part right here, this is the part of collecting duct, collecting tubule. So here this is one of the epithelial cells of the collecting tubule, tubular epithelial cell. Now on the basolateral surface, this, so here there's a receptor for ADH which is called as V2 receptor. Okay, so this is posterior pituitary. Posterior pituitary produces ADH. It and this ADH acts on these receptors, which are V2 receptors. Now these V2 receptors, once it acts on these, they are actually stimulatory type of G protein coupled receptors. They result in formation activation of different proteins, and these proteins eventually uh, they cause expression of certain channels on this surface. These are called as aquaporins, and ADH causes, this V2 receptor causes expression of aquaporin 2. So once there is expression of aquaporin 2, through this aquaporin 2, water can easily pass and there is reabsorption of water. So that's how ADH acts. Again, going back to this diagram, it means if ADH is going to come into action, the result will be net result. Okay, now this is gonna be like opposite. I made, so actually the cell that's shown in this diagram actually belongs to this part of nephron. Okay, so yes, water will be reabsorbed from this side, and this is the other side where V2 receptors are present, and on this side water will be reabsorbed from this side. Okay, this is how ADH acts. Okay, that means if ADH comes, ADH is gonna act on its receptors. ADH acts on its receptors and causes reabsorption of water through AQ2. It is called as aquaporin 2. So net result will be reabsorption of water. But okay, let us imagine there is reabsorption of water. This is reabsorption of water through aquaporin 2. It goes into the medullary interstitium. Now this water in the medullary interstitium, it can again come and go back into this collecting tubule, so it doesn't make a difference. Urine will again be diluted because it can easily. Now the only reason this water has got to stay in this interstitium is if the interstitium has high concentration. If this interstitium is highly concentrated, if this is highly concentrated, only then this water will stay here. If it's not concentrated, this water will enter back into the collecting tubule and be passed out with urine. As a result, there will be dilute urine. This will not be concentrated. So how is the concentration of medullary interstitium maintained so that this water actually stays here? That occurs because of these two components, vasa recta and urea. Okay, now let us try to understand what actually happens here. Okay, I am drawing a rough quick diagram. Okay, so this is a rough diagram. Now, as you all know, this is the descending limb of loop of Henle, and this one here is the ascending limb of loop of Henle. Okay, you all know this. Now, if we talk about the blood supply, okay, this was the efferent arteriole, these were the glomeruli, efferent arteriole. Now, if we talk about vasa recta, it's present exactly in this part, again in U fashion. What is this? This is vasa recta. Okay, this is vasa recta. We all saw what happens in other parts of nephron. Now, in case of vasa recta, things are a bit different. Now, here it goes in this direction, what's called as this direction. That means this limb here is the descending limb, and this one is the ascending limb. Okay, so we all saw that the nephric filtrate here, the concentration was 285 to 300. Here it was again 285 to 300. Here it was 1200. Here it was 50. Now when it has to pass out, its concentration, it should be 800 to 900. Okay, that's the thing. And for that, here there should be reabsorption of water, and that reabsorption of water is going to take place because of ADH. So once ADH comes, it acts on this tubule, and as a result there is reabsorption of water. But the point is that this water, it will not stay in the interstitium. If the interstitium does not have high concentration, this water will otherwise again go back in the collecting tubule. Okay, through aquaporins, there is no specificity for aquaporins. They will only allow water to go out. They can again allow water to enter in if the concentration gradients are reversed. So we need to maintain the osmolarity of this medullary interstitium. So how does that occur? Let us try to understand that. Through the collecting duct, this collecting duct is also permeable to urea. So through this, urea passes out. U is urea. Urea is reabsorbed and it goes into the interstitium. Now this urea is actually responsible for maintaining the interstitium osmolarity. But the question is, this urea again is not going to stay in the interstitium. Maybe this urea will reach into the interstitium, it will spread, it will reach here, it will be urea. But this urea is eventually going to diffuse to, it will be again, once it reaches here, maybe it will be reabsorbed or something, it's not going to stay in the interstitium. So for that, we have this mechanism which is called as counter current exchanger. So what happens, this urea, it is absorbed by the descending limb of vasa recta. Once it's absorbed by descending limb of vasa recta, it again goes down, reaches the ascending limb, and here it's again secreted out. This is urea. Again, from here, it again goes into the descending limb. Okay, so it goes into the descending limb, then as in ascending limb it's thrown out, again reaches into the interstitium. From interstitium, it again goes into the descending limb. So because of this exchange thing, it goes into the descending limb, then through the ascending limb, it again goes into the interstitium, then goes into descending limb, ascending limb, interstitium. So this helps to maintain the concentration, the osmolality of interstitium to a high value. So it's because of urea and because of vasa recta, and because of this thing, this water is going to diffuse out on action of ADH. So once ADH acts, this water will diffuse and will stay in the interstitium till it be reabsorbed because of presence of urea. And presence of urea in the interstitium will be because of countercurrent exchanger. And for countercurrent exchanger, vasa recta is required. Okay, so this is the counter current exchanger. Vasa recta is required for this, and urea is also required for this. And obviously we require ADH. So once this countercurrent exchanger works, the osmolarity of medullary interstitium is high. Okay, this is high. There will be reabsorption of lots of water, so water will be reabsorbed, and the concentration from 50 it's going to increase till it forms urine. It's going to become 800 to 900, and as a result we are able to excrete what's called as a highly concentrated urine. So this is how the counter current exchanger works. I hope you all understood it. If there's some doubt, you can write down in the comment section. Uh, if I fail to explain some part of the video clearly, please write it down in the comments. I'll try to make a new video uh, trying to explain that part better. So I hope you got it all, and thank you.